Very good afternoon to all the students. So, yesterday we are done with photosynthesis in higher plants first session, connecting to the 10th biology first chapter, that is nutrition, where we have learned the complete framework of this chapter, photosynthesis in higher plants, highlighting both light reactions and dark reactions. 3 steps of light reactions we have done in detail. The very first step, absorption of light in the form of photons. Second is photolysis of water. Third, assimilation of energy, assimilatory power, creating assimilatory power in the stroma of the chloroplast, that is formation of ATP and NADPH. Now, utilizing this ATP and NADPH, glucose is fixed from carbon dioxide. Okay? By using the assimilatory power, glucose is fixed from carbon dioxide. So, yes students, if we have to observe, yesterday we have learned the complete framework and we have also discussed about chloroplast organelle, third membrane system, that is thylakoid membrane. Inside thylakoid membrane we have learned there are light harvesting complexes. So, once if we have to recall, if I have to take this a thylakoid sac, one thylakoid sac, this is the membrane of thylakoid which is addressed as thylakoid lamellae or thylakoid membrane. Inside the thylakoid lamellae or membrane, we have light harvesting complexes which are known as photosystem 2 and photosystem 1. The absorption maxima of photosystem 2 is 680 nanometers and the absorption maxima of photosystem 1 is 700 nanometers. We have also discussed that each photosystem will be having both core pigment and assimilatory pigments, assimilatory or antenna pigments, assimilatory or antenna pigments. Clear, right? These are present in photosystem, in either of the in both of the photosystem 2 and photosystem 1 we have core pigment and accessory or antenna pigments. Accessory or antenna pigments can be chlorophyll B, carotenoids and xanthophylls. Very important, remember, because if you have clear idea on pigments and their absorption spectra, then only you can answer any question which is coming from action spectra, absorption spectra that we have exercised yesterday. Pigments are very basic to understand. Okay. So, this accessory or antenna pigments, what do? They will absorb light at different wavelengths from 400 nanometers to 600 nanometers. Okay. They will absorb the light at different wavelengths from 400 nanometers to 600 nanometers, not at the highest wavelengths. So, how the sunlight is absorbed? I told you sunlight is absorbed in the form of units, those are photon units. Absorbing the energy, the electrons get excited from the pigment molecules and the excited energy is transmitted to the primary acceptor. Primary acceptor is chlorophyll A and the absorption maxima of this chlorophyll A in photosystem 2 is 680 nanometers and the absorption maxima of chlorophyll A in photosystem 1 is 700 nanometers. Okay. So, these are present where in thylakoid membrane. Fine, right? Once the energy is excited to the reaction center, that is chlorophyll A, the energy is transmitted to the lumen, that is utilized to split the water molecule inside the lumen. Inside the lumen, photolysis of water happens. Lysis means what, breakdown. Photo means using the energy, that is physical energy in the form of photons. Utilizing that energy inside thylakoid lumen, water molecule is breaked to 2 protons plus 2 electrons and oxygen molecule. Got it? Now, here the electrons, protons which are formed inside the lumen. So, in thylakoid lumen, if you observe keenly, towards the inner side of the thylakoid membrane here, towards the inner side, there is a complex called as oxygen evolving complex is present. Okay? Towards the inner side of the thylakoid membrane, towards lumen, oxygen evolving complex is present. This complex utilizes the energy from photons and it breaks or lyses water molecule into protons, electrons and oxygen molecule. Okay? So, protons are present inside the lumen, whereas electrons get excited. Okay? Electrons get excited. Protons are present inside the lumen, but electrons get excited into the photosystems. Here if you observe, yesterday we didn't discuss about non-cyclic photophosphorylation, but non-cyclic photophosphorylation is very important. Now, we are going to learn the third step of light reaction. First step is clear to us, how the absorption of light in the form of photons takes place. How the absorption take place ma? Through light harvesting complexes, that is photosystem 2 and photosystem 1, which are present in the thylakoid membrane, which consists of pigments. Accessory or antenna pigments will absorb light at diverse wavelengths from 400 nanometers to 600 nanometers, and then energy gets excited, which is accepted by the primary acceptor. Remember, primary acceptor is chlorophyll A. Chlorophyll A in photosystem 2, maximum absorption is 680 nanometers. In photosystem 1, chlorophyll A maximum absorption is 700. Likewise, first step of light reaction is clear to us. Second step, by oxygen evolving complex, which is present on the inner side of the thylakoid membrane towards lumen, where water molecule is photolysed to 2 protons, 2 electrons and oxygen atom. First two steps are clear to us. Now, third step, which is formation of assimilatory power. Third step is formation of assimilatory power, which is very important. Listen with much focus, interest and enthusiasm, because formation of assimilatory power, that is ATP and NADPH. Formation of ATPs in higher plants is learnt under the concept non-cyclic photophosphorylation. This is learnt under the concept non-cyclic photophosphorylation. Just a second. So, here we have understood that over the thylakoid membrane photosystems are present and inside the lumen water molecule is split. Now, here electrons get excited in the photosystems. How these electrons are excited, we are learning here. Okay? Assume this as thylakoid lumen and this is thylakoid membrane. Okay? First, electrons get excited to the reaction center of photosystem 2, PS2. So, the reaction center of PS2, absorption maxima of chlorophyll A is P680, that is 680 nanometers is the absorption maxima, right. From here, the electrons are first accepted by 2 electrons are first accepted by pheophytin. Pheophytin. Remember, this is the primary electron acceptor which is present on the outer side of the thylakoid membrane. Next, phaeophytin will submit the 2 electrons to plastoquinone. Remember, plastoquinone is also a proton acceptor. This is what side ma? Stroma side. So, from the fluid in the chloroplast, that is in the stroma, whichever protons are present are transmitted or passed to the plastoquinone. Phaeophytin is submitting 2 electrons to plastoquinone. At the same time only, PQ is taking protons from the stroma and this gets converted to plastohydroquinone, PQH2. And from here, protons are submitted. From here, protons are submitted to lumen and electrons are pumped to lumen and electrons are pumped to cytochrome B6F complex. From cytochrome B6F complex, the last electron acceptor of photosystem 2 will be plastocyanin. Got it right? Clear all? Very important to remember this electron flow. This is thylakoid lumen. Yeah. Listen all carefully. So, here in photosystem 2, the electrons first get excited to chlorophyll A, which is the primary acceptor at the reaction center. And here the absorption maxima is P680. From here, the electrons are first given to phaeophytin. Phaeophytin is the primary electron acceptor. From phaeophytin, the electrons are given to plastoquinone. Plastoquinone parallelly accepts the protons from stroma and converts to plastohydroquinone, where protons are sent to the lumen, electrons are given to cytochrome B6F. Okay, these terms might sound new to you, but get adapted to it so that it will be easy for you when we are starting photosynthesis in the new academic session. Okay, try to assimilate this. Okay, don't memorize now only, but try to assimilate the electron flow. Okay, so here protons are submitted to lumen and electrons are given to cytochrome B6F. From here, electrons are again given to plastocyanin. So, first reaction from here thylakoid lumen to the reaction center is uphill movement, and then this electron flow is downhill movement. What is the primary electron acceptor of photosystem 2? Phaeophytin, then plastoquinone, then cytochrome B6F, then plastocyanin. Plastocyanin is the last electron acceptor of photosystem 2. Remember all these statements carefully, and the electron flow is very important. And then here, if you look into from plastocyanin, the electrons get excited to photosystem 1. Photosystem 1, where the maximum absorption or the absorption maxima is P700. P700 is the absorption maxima here. So, if you observe here the electrons again move in the uphill direction. First excitation is uphill, then electron flow in PS2 is downhill, and again from here uphill to PS1. Here PS1 what is accepting? Ferredoxin. Ferredoxin is accepting the electrons in photosystem 1. From ferredoxin, the electrons are submitted to ferrous sulfur complex. From ferrous sulfur complex, electrons are given to the ultimate electron acceptor, that is NADP+. NADP plus is present in the stroma side of the thylakoid membrane. Stroma side. Okay, wherein NADP plus is reduced to NADPH plus H plus. This is by NADP reductase. This is by NADP reductase enzyme. Got it? Clear? So, once again let us have a quick review on non-cyclic photophosphorylation. At the end of non-cyclic photophosphorylation, what is formed, ma? NADPH is formed. Non-cyclic photophosphorylation is operated in thylakoid membrane by two photosystems, which are the light harvesting complexes. Each photosystem will be having core pigments and accessory pigments. Absorbing light in the form of photons, the water molecule is split in the lumen and it gives to protons, electrons and oxygen atom from single water molecule. Okay. Here electrons get excited to the reaction center and two electrons from the reaction center are accepted by the primary electron acceptor, that is pheophytin, followed by plastoquinone. Plastoquinone parallelly takes protons from the stroma, converts to plastohydroquinone, then protons are submitted to the lumen. And here from plastohydroquinone electrons are given to cytochrome B6F complex and then electrons are given to plastocyanin. From here two electrons are given to photosystem 1 where the primary electron acceptor is ferridoxin. From here ferrous sulfur complex, ultimate electron acceptor is NADP plus. NADP plus is present on the stroma side of the thylakoid membrane, on the stroma side. And here to reduce NADP plus, we have the enzyme that is NADP reductase, wherein NADPH plus H plus are formed. But then in the assimilatory power, only NADPH is formed here, we still yet didn't form ATP. How ATP is formed is proposed by another hypothesis, that is chemiosmotic hypothesis, okay, which is learnt under chemiosmotic hypothesis. Remember children, in every year NEET examination, either from non-cyclic photophosphorylation or from chemiosmotic hypothesis, there will be one question, definitely. So, listen it carefully, try to assimilate. Memorizing part we will do it later, okay. Try to assimilate with the terms, try to assimilate with the proton channels, try to assimilate with the flow of the electrons. Here in chemiosmotic hypothesis, 3 points you have to remember. The very first point is, because of photolysis of water inside the thylakoid lumen, where water molecule is oxidized to electrons and protons. Likewise, the lumen is rich with protons, that is first point. Thylakoid lumen, first point is rich with protons because of photolysis of water inside the lumen. This is the first source of protons. Second point, in the non-cyclic photophosphorylation we have learnt. Here it is taking the protons from stroma, right? Plastoquinone is taking the protons from stroma. These protons are again travelling from stroma to lumen, okay. From here plastohydroquinone is submitting to lumen. Again protons via plastoquinone, when it is accepting protons, this is being converted to PQH2 and then protons are submitted to lumen. This is the second step. Again from stroma to lumen we are getting protons. See here from this diagram. 2 steps where lumen is rich in protons than stroma. Third point is the last step of non-cyclic photophosphorylation, whichever little amount of protons are present in the stroma, those protons are accepted by NADP plus. Those protons are accepted by NADP plus, and NADP plus is converted to NADPH. Okay. What is the full form of this, ma? Nicotinamide adenine dinucleotide hydrogen phosphate, wherein NADP is reduced to NADPH. Likewise, protons are completely reduced in the stroma. So, third step for reduction of protons in stroma is NADP plus is reduced to NADPH. Got it? So, this is reduced. Likewise, a proton gradient is generated from stroma to lumen. In stroma, completely proton concentration is reduced, whereas in lumen there is high proton concentration. Now, to attain equilibrium, the protons now we need to pump from which direction to which direction, ma? From lumen, protons have to get into the stroma so that they will attain the equilibrium, right? For this, and a channel is activated in the thylakoid membrane, that is known as ATP synthase complex. Okay, that channel name is what, ma? ATP synthase complex, or it is also addressed as F0, F1 particles. These are also addressed as oxysomes or Fernandez-Morgan particles. Fernandez-Morgan particles. This has these many synonyms. Most of the times in your questions, it will be asked either with the name of ATP synthase or with the name of F0, F1 particles or oxysomes mostly. So, remember these three for sure. Here F0, F1 complex is there, right? F0 is embedded inside the membrane, F1 is towards the stroma. In a single run, F0, F1 complex will pump 4 protons from lumen to stroma. Okay. So, if you consider previously, in stroma proton gradient got reduced because of these 3 reasons. Very important, you have to remember these 3 reasons. That is the reason you have to know all the 3 steps of light reaction. First reason is photolysis of water, because of which protons are rich in lumen. Second reason is protons are pumped from stroma to lumen via plastoquinone. Third is protons are reducing NADP plus, H plus is reducing NADP plus to NADPH, where protons reduced in stroma, increased in lumen, pH is low in lumen and high in stroma. So, because of this proton gradient, a channel is activated which is known as F0, F1 channel, which is also known as ATP synthase or oxysomes. This will pump the protons in a single run. How many protons are pumped? 4 protons are pumped. This 4 protons will phosphorylate ADP plus inorganic phosphate into ATP, adenosine triphosphate. Inside stroma, whichever adenosine diphosphate and inorganic phosphate are there, this ADP will undergo phosphorylation through protons, consuming the energy from protons. Likewise, 4 H plus are required to form 1 ATP. So, now you understood how the assimilatory power is formed, that is ATP and NADPH via non-cyclic photophosphorylation and chemiosmotic hypothesis. Clear, right, ma? Now, so now you tell me what is the site of this reaction where ATP and NADPH are formed. Remember very carefully, ATP and NADPH are formed in the stroma of the chloroplast. Stroma of the chloroplast. Okay, remember this. So, now in the stroma of the chloroplast, we have the complete energy which is required for the formation of glucose. Now, we are ready, all set for the formation of glucose. Now, how this will happen? Listen carefully. Before getting into the dark reactions, those are light independent reactions. In that I told you about 3 cycles, that is C3, C4 and CAM cycles. I will give you a complete idea on all the 3 cycles and we will discuss whole and soul on C3 cycle. Okay ma. Before going to that, let us exercise one NEET PYQ which is asked on chemiosmotic hypothesis in NEET 2023. Look at it carefully beta, look at it carefully and answer. Try to answer it, even if it is wrong, no problem, but try to answer. Yeah, let's get into the question. Which of the following combinations is required for chemiosmosis? Okay, what are requirements of chemiosmosis? Now you tell me, looking into the question carefully, we need proton gradient, ATP synthase, membrane and F0 F1 complex. So here look at the options and answer carefully ma. We need a membrane, a proton pump, proton gradient and ATP synthase complex, which is also known as F0 F1 complex. So what is the right answer? Option 1. Clear, right? So now we are getting some confidence that you can address all the NEET questions. Okay. You need to go step by step. In a single class or in a single day you will not understand everything, but then you will be confident that you are achieving the first step, you are assimilating and understanding all the concepts. That confidence and that boost up you will get in the beginning of your journey. Okay, but don't get disheartened if you are unable to answer. Okay, at least if you are understanding the concept, go on that positive note. Understand the objective. Okay, this likewise we need to go through the objective pattern. Okay, slowly try to inculcate objective oriented learning. Okay, and now let us move to light independent reactions. Light independent reactions. Also known as dark reactions. Light independent reactions, also known as dark reactions. So here look at it carefully and answer. Calling light independent reactions as dark reactions is a misnomer, because what does the term dark reaction means? It means that these reactions are occurring only in the dark. It doesn't mean that. Light independent reactions mean it is dependent on the products of light reaction. On what products it is dependent on? On ATP, on NADPH it is dependent on. It is not dependent on the light directly, it is dependent on the products of light reaction. That is the reason it is addressed as light independent reactions, also known as dark reactions. So here if you observe C3 cycle, C4 cycle and CAM cycle. C3 cycle, only C3 cycle is operated in C3 plants or temperate plants. Okay, only C3 cycle is operated in temperate plants. Okay, this we have discussed in the beginning as well. C3 cycle is also addressed as Kelvin cycle, which is discovered by Melvin Kelvin. The cycle is named after the scientist who has discovered this. It is discovered by Melvin Kelvin. How did Melvin Kelvin discover the C3 cycle? He has discovered it through a radioactive isotope, that is C14. C14 is a radioactive isotope. Using this radioactive isotope on chlorella, which is unicellular green algae, he has conducted algal photosynthesis. When he has conducted algal photosynthesis using C14 radioactive isotope on chlorella, he observed that the first stable organic intermediate formed is 3-phosphoglyceric acid. The first stable intermediate formed is what, ma? 3-phosphoglyceric acid, which is a 3-carbon compound. That is why he named it as what? C3 cycles. C3 cycle occurs in all the photosynthetic plants. Like in temperate plants, only C3 cycle occurs. In tropical plants, along with C3 cycle, C4 cycle happens. In CAM plants, along with C3 cycle, CAM cycle happens. Okay, but then C3 cycle occurs in all the photosynthetic plants. Only C3 cycle without any other cycles operates in temperate plants. I hope you got the point, right? So, the first stable intermediate formed is 3-phosphoglyceric acid, that is why it is named as C3 cycle. Now, let us learn the intro of, now let us, yes ma, you can ask your doubt, you can ask your doubt. C4 cycle is also known as Hutchinson-Slack pathway. Yeah, you ask your doubt, ma. I will address it. You type the doubt you have. Meanwhile, we will go through C4 cycle. Okay. Algal photosynthesis. Okay. Listen, ma. Algal photosynthesis is nothing but the photosynthesis which occurs in algae, and that is oxygenic photosynthesis, not anoxygenic photosynthesis. Oxygenic photosynthesis. When we are discussing about types of photosynthesis yesterday, we have discussed about 2 types of photosynthesis. One is oxygenic, one is anoxygenic. In anoxygenic photosynthesis, the electron donor and proton donor will be H2S. Likewise, the element sulfur is formed as the byproduct in bacterial photosynthesis. Anoxygenic photosynthesis, bacterial photosynthesis can be used as synonyms. Likewise, in algae, which are the primitive plants, we call it as under the thallophyte division. In algae, oxygenic photosynthesis occurs, wherein water is the electron donor and this forms the oxygen. Oxygen is evolved as a byproduct from the electron donor and proton donor, that is water molecule. That is the reason it is known as oxygenic photosynthesis. As it occurs in algae, the term is given as algal photosynthesis. No, all the algae will be performing the algal photosynthesis. Radioactive isotope is nothing but whenever we are conducting any experiments in the laboratories, whenever the experiments are done, we can take the isotope of carbon. Okay, one of the isotope of carbon is C14. Okay, likewise, if you see in Michelson and Stahl's experiment, okay, isotope of nitrogen is taken as N15. Okay, heavy isotope we will be calling it as. So, whichever elements we are using in the laboratories to conduct the experiments, their isotope, carbon isotope is taken as C14 by Kelvin. Got it? And he has performed algal photosynthesis on chlorella, where he has discovered the primary product formed is 3-phosphoglyceric acid. Now, you got the point, ma? If you are having any other doubt also, you can freely ask. There is no problem in that. Okay. Meanwhile, go through C4 cycle, which is also known as Hutchinson-Slack pathway. Here, it is known as C4 cycle because the first stable product formed here is oxaloacetic acid, which is 4-carbon organic acid. So, here first stable carbon or the first stable intermediate formed is 3-carbon. First stable intermediate formed here is 4-carbon. Why is it so? You will get to know when you are learning the first step in these 2 cycles, that is carbon fixation. As of now, you remember in this introductory session that in C4 cycle, the primary product is oxaloacetic acid, which is 4-carbon compound. Very good, ma. Very good. You got it right. Continue with the class, beta. Malic acid and aspartic acid. These are the 4-carbon organic acids which are formed. This occurs only in C4 plants. You can see C3 cycle occurs in all the photosynthetic plants. C4 cycle occurs only in the C4 plants. Clear, right? C4 plants are those plants which grow in tropical climates. You can also address it as tropical plants. Most of the monocots, most of the monocotyledonous class will be conducting C4 cycle itself. You can take the examples as sorghum, maize and sugarcane. There are few exceptions from dicots which will also conduct the C4 cycle, for example amaranthus. Clear? So, what is the introductory we have learnt from C4 cycle? C4 cycle also known as Hatch and Slack pathway, where the first stable product formed is oxaloacetic acid. It can convert to malic acid and aspartic acid also. C4 cycle occurs only in C4 plants which grow in tropical climates. Mostly occurs in monocots, for example sorghum, maize, sugarcane. It also occurs in few of the dicots as exception, for example amaranthus. Sharma will arrange that. We will arrange the session very soon, okay. For atomic structure revision, we will be doing it, okay. It is noted and you will get the session very soon. Now, CAM cycle, okay. CAM cycle is crassulacean acid metabolism. The full form of CAM is crassulacean acid metabolism. This occurs in crassulacean family members, okay, and here also the first stable product formed is oxaloacetic acid. It is known as CAM cycle because it occurs mostly in what plants? The plants which are belonging to which family? Crassulacean family, okay. This occurs in those plants which are in what climates? Desert climates. C4 cycle occurs in what climates? Tropical climates. So, here the plants are known as succulent plants, for example cactus, agave and opuntia, aloe vera, these are the plants, okay. Tropical epiphytes for example orchids, okay. These are the plants in which CAM cycle is operated, and this is about C4 cycle, this is about CAM cycle, also known as crassulacean acid metabolism. First organic stable product is same as that of C4 cycle, that is OAA, but then the CAM cycle is named because this occurs only in crassulacean family members. You will know when we are learning the biosynthetic pathways in detail, you will know the clear difference between C4 cycle and CAM cycle. This occurs in these plants, succulent plants and tropical epiphytes. Clear, right ma? Till here I hope everything is clear. Now, we are going to learn whole and soul complete biosynthetic pathway which is happening in C3 cycle, which is very important for your NEET and MCET, okay. Only C3 cycle we are going to learn in detail today, okay, fine ma. Let us learn it. C3 cycle is operated in 3 steps. Listen with utmost focus and attention, it is very important to be known. So, first step is carboxylation, second step is known as reduction, third step is known as regeneration. These are the 3 steps which occur in C3 cycle. So, first step which is carboxylation, this is known as most crucial step of Kelvin cycle, okay. Carboxylation is known as what? Most crucial step of Kelvin cycle. Via stomata, listen, via stomata, the carbon dioxide is accepted into the plant. It enters into the mesophyll cell and then enters into the chloroplast. In chloroplast stroma, carbon dioxide is landed. Now, this carbon dioxide should be accepted by some molecule, right. So, the primary acceptor of carbon dioxide is RUBP, which is ribulose bisphosphate, okay. After carbon dioxide is being accepted by RUBP, which is 5 carbon compound, this is catalyzed by Rubisco, which is ribulose bisphosphate carboxylase oxygenase. Remember students, the name itself is saying that it has dual functions of carboxylase and oxygenase. Rubisco, ribulose bisphosphate carboxylase oxygenase. Okay, it performs both carboxylase function and oxygenase function. Remember here one point I will tell you, Rubisco will perform dual functions, right? In the atmosphere, for C3 plants, if there is equal concentration of carbon dioxide and oxygen, Rubisco will perform carboxylase activity only. But oxygen concentration is more, carbon dioxide concentration is less, then Rubisco will perform oxygenase activity. This is one major drawback, okay, of this enzyme. And remember this point also, Rubisco is known as world's abundant enzyme. Okay. So, this performs oxygenase activity when oxygen concentration is more and carbon dioxide concentration is less, whereby, see, carbon dioxide has how many carbons? 1 carbon. I told you RUBP is having 5 carbon compound, total 6 carbons, right? So, 2 molecules of 3 carbon compound are formed as a primary organic intermediate, and that is 3-phosphoglyceric acid. Got it, ma? 3-phosphoglyceric acid is formed. How many molecules? 2 molecules of 3-phosphoglyceric acid are formed when 1 carbon dioxide is fixed, because it is 3 carbon compound. Now, the second step I spoke about is reduction. In reduction, 3-phosphoglyceric acid should be reduced to glyceraldehyde 3-phosphate. Okay, 2 molecules of G3P is reduced to 2 molecules of glyceraldehyde 3-phosphate. For this, we need 2 molecules of ATP and 2 molecules of NADPH as the energy source to reduce PGA into G3P. Now, 2 molecules of glyceraldehyde 3-phosphate, on whole it has how many carbons, ma? 6 carbons, right, of which 1 carbon will be leaving the cycle for the formation of glucose. Ma'am, where this 1 carbon is going? It leaves into stroma itself, it is leaving the cycle, it is leaving this cycle and moving to stroma. Okay. So, here this 1 carbon is moving away when 1 carbon dioxide is fixed. Likewise, in every cycle, 1 carbon dioxide is moving into stroma for the formation of glucose. We know glucose has how many carbons? 6 carbons. Likewise, how many carbons should leave the cycle? 6 carbons. Means 6 carbon dioxides have to fix, 6 Kelvin cycles have to happen for the formation of 1 glucose. Got it, ma? So, now from the 6 carbons, when 1 carbon is leaving the cycle, we have rest how many carbons? 5 carbons are there. The rest 5 carbons will be regenerating RUBP, which is the third step of Kelvin cycle. For this regeneration step, it requires again 1 ATP. Got it, right? So, for the regeneration, from the 6 carbon, 1 carbon will be leaving for the formation of glucose, rest 5 carbons will be forming what? RUBP. Okay. This step is known as regeneration, where for this regeneration step 1 ATP is required. Regeneration step is addressed as most crucial step to continue the cycle uninterruptedly. So, here what points you have to take at the end of Kelvin cycle session? 1 carbon dioxide will form 2 molecules of 3-phosphoglyceric acid. For this reduction, 2 ATP and NADPH are required. From this, 1 carbon is leaving the cycle for the formation of glucose, rest 5 carbons are regenerating back for the continuation of the cycle. Likewise, for the formation of 1 glucose, how much energy is required? For the conducting of single Kelvin cycle, how much energy is required? We need to address the bioenergetics. Bioenergetics are very important for C3 cycle and C4 CAM as well. But now today we learn C3 cycle, right? So, here bioenergetics of C3 cycle. Okay. For 1 Kelvin cycle to occur, it requires 1 carbon dioxide, right ma? And how much ATP? 2 ATP here in the reduction, 1 ATP for regeneration. So how much ATP is required? 3 ATP on total. How much NADPH is required? How much NADPH? 2 NADPH are required, right? 2 NADPH. But our ultimate goal is what? Formation of glucose. For formation of one glucose molecule, here you see, from each Kelvin cycle, 1 carbon is escaping for formation of glucose. Likewise, how many Kelvin cycles have to happen? 6 Kelvin cycles. For this, how much carbon dioxide is required? 6 carbon dioxide. How much ATP is required? Just to multiply 3 into 6. Total how much ma? 18 ATP. Got it? 2 NADPH. How much NADPH is required? 2 into 6. We have 12 NADPH. 12 NADPH. Got it? So here for the formation of one glucose molecule, 6 Kelvin cycles, 6 carbon dioxides, 18 ATP and 12 NADPH are required. So if you are quite confident with the Kelvin cycle, let us face the NEET PYQs. This is a question which is asked in the NEET 2025 on Rubisco. Try to answer this ma, everybody try to answer this. Even if it is wrong, no problem, but try to answer. Okay. Look at it. Which of the following statements about Rubisco is true? We need to identify the correct statement. It is an enzyme involved in photolysis of water? No. For photolysis of water, which is happening in the lumen of the thylakoid, there is no enzyme required. It requires only oxygen evolving complex. Got it? Next, it catalyzes the carboxylation of RUBP, correct, which is the first step of Kelvin cycle. Got it? Next, it is active only in the dark? Wrong. It is not active only in the dark, it is active during day also. It has high affinity for oxygen than carbon dioxide? Wrong. It has equal affinity for carbon dioxide and oxygen. So what is the right answer? Option 2. Got it ma? Get exposed slowly to the questions, you will get into the track very soon. Second question, which is asked in NEET 2024 on the energetics of C3 cycle. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in the Kelvin cycle? So, for one carbon dioxide molecule, that means for one Kelvin cycle, how much ATP and NADPH are required, he is asking. We have learnt clearly that in the second step of Kelvin cycle, that is in reduction, it requires 2 ATP and 2 NADPH. Third, for regeneration, it requires 1 ATP. So, on total, how many ATP are required? 3 molecules of ATP, 3 molecules of ATP and 2 molecules of NADPH, because NADPH is required only for the step of reduction. So, what is the right answer here? Right answer here is option 4. Got it? Right answer is option 4. Now, the last question of today's session, again on the energetics of Kelvin cycle, asked in the year NEET 2023. Try to answer, no problem. We have learnt it, right? Just now we have learnt it. Even if it is right, wrong, no problem. Try to answer. The question is, how many ATP and NADPH are required for synthesis of one molecule of glucose? We have learnt very clearly that one molecule of glucose is formed when 6 carbon dioxides are fixed, right? When 6 Kelvin cycles are operated. For this, it requires how much carbon dioxide? 6 CO2, which means 6 into 3, 3 ATP for 1 CO2, right? So, 6 into 3, that is 18 ATP, and 6 into 2, which is 12 NADPH. Okay, 6 into 2, which is 12 NADPH2. So, here the right answer will be 18 ATP and 12 NADPH2. Okay. So, to all the students, these 2 sessions where we have learnt in photosynthesis in higher plants. Key takeaways I will give you. In the nutrition chapter, which is the very first chapter from your biological sciences of unit of biological sciences of chapter 1 nutrition, where you will learn just on the discoveries, okay, and the very basic steps of light reaction, and you will be learning an introductory paragraphs of dark reaction, that is all. That a statement that glucose is formed. But here in your NCERT 11, you will learn in detail about the 3 steps of light reaction, okay, where photolysis of water is happening, how the light is absorbed as photons, by what photosystems, what are the pigments involved, what will be the absorption range of that pigments and photosystems, and at last how ATP is formed via non-cyclic photophosphorylation, how NADPH is formed, okay. All this you will be learning, how ATP, NADPH are formed, 2 cycles you have to remember, non-cyclic photophosphorylation and chemiosmotic hypothesis, which results in the formation of ATP. And then for the formation of glucose, what kind of plants will operate what cycles? Temperate plants only C3 cycles, tropical plants C3 cycle and C4 cycle, desert plants C3 cycle and CAMP cycle, of which today we have learnt whole and soul completely about C3 cycle, okay, where we have learnt 3 steps: carboxylation, reduction and regeneration. You have to keenly focus on what is the primary acceptor, what is the enzyme that is required for carboxylation, how much of energy is required to fix 1 carbon dioxide, how much energy is required for the formation of 1 glucose, because you will get questions from enzymes, from bioenergetics and substrates or organic intermediates, okay. And finally, we have solved 3 to 4 NEET PYQs, 1 PYQ on chemiosmotic hypothesis, 3 PYQs on C3 cycle itself, okay. So, with this we have completely done with the chapter of photosynthesis in this bridge course. Tomorrow we will be moving into respiration, okay. Thank you.